Hello, GrainGenes user, and thanks for watching this tutorial. At GrainGenes, we've been curating genetic and genomic data for the small grains for nearly 3 decades. But data is published faster than we can curate it, and we need your help. We have several ways to submit data, and a good start is by clicking the Submit Your Data to GrainGenes link on the homepage. Like the images suggest, we can add genome browsers de novo and tracks to existing browsers, image collections, genetic maps to use in the comparative mapping tool CMAP, and germplasm data, new marker sets, etc. Click on the Excel template link to download the workbook to pre-curate data for GrainGenes. When you open the workbook, you should be at the Introduction tab. From the table of data types, you can link to the instructions in the All Data Types tab or link directly to the data templates. Notice the templates are also the tab names at the lower part of the screen. The All Data Types page provides examples and notes for the most used data tags. The data type link in the header row for each data type navigates to the template page. And from the template, the data fields are links that go back to the All Data Types page. In the first example, I'll demonstrate how to submit new maps to GrainGenes. Many of the oat genetic maps in GrainGenes were pre-curated by Charlene White of AAFC Canada. Those include maps from this publication mapping a small rust resistance gene out of Kathy Close's lab. In this publication, the authors report the crown rust resistance gene PC53 in 2 populations. The gene maps to the MERG8 linkage group in both populations, along with a few dozen SNPs. Okay, so here's the workbook that Charlene submitted to GrainGenes in 2020. Notice that she renamed the workbook to a descriptive name prior to sending it to us. And here, GrainGenes only needs the supporting information for this paper, which includes the reference, germplasm, gene, map, and map data templates. Since there are no new QTL, loci, probes, or sequences, she can just leave those pages blank. To reiterate this, since there are already complete locus records for the SNPs on these maps in GrainGenes, she did not need to repeat that information here. We will discuss more of the curation process in the next example, but here is the outcome. A map data record holds the metadata for a map set and links to the germplasm records for the parents, the reference, some kudos, and a list of loci. Clicking on a map name will render a map in CMAP. The example shows the PC53 by PC50 map aligned with the 2018 OAT consensus map. This is detailed in the tutorial called Using CMAP to Enrich Markers Around a Gene of Interest and is available in the GrainGenes collection at YouTube. In this example, we'll demonstrate how to submit GWAS or QTL data to add to a map that already exists in GrainGenes. In this case, the 2015 integrated map from Macaffery et al. The integrated map from Macaffery et al. Was first curated in 2015 and is available as wheat, YR, genes, and QTL. In 2021, the map was re-curated as wheat, YR Update 2021 with the following updates. Chromosome extensions were added to locus names that were found in GrainGenes mapping to more than one locus. Some QTL in the paper were curated in 2015 as single position loci. These were placed on the updated maps to reflect the entire span. And map data was available from the original publications as centimorgans and relative position on the chromosome. Wheat YR genes in QTL was curated with the relative position. The updated maps are drawn in centimorgans. In a project launched in 2021, QTL results measuring stripe rust resistance from several wheat collections are being added to the 2015 integrated map from Macaffery. The next few slides will demonstrate the addition of the results from this 2020 publication. The next two slides show the QTL tables indicating the significant marker, stats, and the resistance-associated allele. These were the race-specific seedling QTL. And these are the adult QTL in different environments. Data from the tables are then added to the appropriate templates. Once data is submitted to GrainGenes, a curator will review the data. In this case, the QTL names fit well with the existing nomenclature. Marker names were checked and chromosome extensions are added for those mapped on more than one locus. Marker positions from the integrated map are then looked up and added to this record. As a result of curation, the reference report will have links to the map data and all QTL records. QTL from this paper can be searched using a truncated form of the locus name. A file assigning QTL to map positions on the wheat YR Update 2021 is written and loaded in GrainGenes. At that point, it can be loaded onto CMAP. These four maps show chromosome 2b throughout the addition of QTL found by GWAS from 4 additional papers on stripe rust resistance. Thanks, and we look forward to hosting your data on GrainGenes.